Okay, so welcome to the next video. As usual, there are some sort of directed revision questions you can have a go at if you feel confident to answer, or even if you feel underconfident, have a go, pause, and then return and I'll discuss the answers. Okay, so what happens to a helper T cell after binding to the antigen on a presenting cell? Well, I just want to take a step back. So in the last video, we talked about what happens with an antigen presenting cell. So I'm showing you an antigen presenting cell here. It's a phagocyte that's taken up, engulfed pathogen by phagocytosis. It's hydrolyzed that pathogen. Lysosomes have fused with the phagosome to release enzymes that have allowed it to hydrolyze and destroy the pathogen. And then what it's done is it's presented some antigen on its surface, making it an antigen presenting cell. In your blood, in your body, there are going to be lots of different types of T cell, and they're different because they have different receptors on their surface for different antigens. They exist from birth. You have always had them. You've never had to be infected with the pathogen. They just are created as part of your development. So you have, I'm showing 4 different types here, but you've got approximately 10 million, it's thought. So one of these, T lymphocytes in blue, has a receptor that matches that antigen. So somewhere in your body, you will have a helper T cell that has a receptor that fits exactly the antigen that is presented on the surface of a antigen presenting cell. Let's say it's this one. That will bind. As we talked about in the last video, that is the link between the non-specific immune system, the phagocyte that becomes the antigen presenting cell, and the lymphocyte, the helper T cell, the helper T lymphocyte, which is part of the specific immune system. It's specific because it has this specific receptor that fits only one antigen. So that's that. That's the activation step. What happens? So to answer question 1, this is what happens. It divides by mitosis, and they divide by mitosis. So you produce more of these helper T cells that have this specific receptor. They're clones. These are activated clones, so they're ready to do several jobs, and they can do one of 4 things. So they're going to do one of 4 things. First thing, so they release chemicals, and you don't need to know this, but they're called cytokines. And the first thing, let me move that because it's not quite what I want you to know, but the first thing they do is, because they release those chemicals, they activate phagocytes to carry out phagocytosis. So that's one of the first things they do. They stimulate that non-specific immune response to keep going. Second thing that you should have. They form memory helper T cells. So they remain in the blood ready for an infection with that same pathogen. So you have a lot more of the helper T cells with that receptor in your blood, so you're ready to respond quickly the next time you're infected. Third thing, they activate the other cells of involved in cellular immunity, which are cytotoxic T cells. We'll talk about those in the next video. The final thing they do the final thing they do is to activate B cells. So there's the connection between cellular immunity and humoral immunity. Remember, B cells are involved in humoral immunity and release antibodies, and we'll talk about those in another video. So that's it, a summary of helper T cells and their function and what they do. So in the next video, we'll deal with the other cells involved in cellular immunity. They are cytotoxic T cells.